Well, thanks for the award. I'm super pleased to be here. I'm going to go ahead and come down here so I can interact with you guys a little bit more while I give this talk. So, over the past several decades, there's been increasing evidence that evolutionary change can happen over ecological timescales, short timescales, not glacial, slow timescales that we normally think of in evolution. If this is true, then this sets up the possibility where ecology and evolutionary processes are interlocked into a dynamic cycle where evolutionary change influences ecological processes, and ecological processes, those changes, then influence processes of evolution. Now, we've known for a really long time that ecology sets up evolution through natural selection, but more recently, there's been increasing evidence that short-term evolutionary changes actually influence short-term ecological dynamics. And there's been a few good papers that have come out on this in the last 5 or so years. But most of them have focused on the net effects of evolution, so how evolution influences the community through all the different pathways that it could possibly be there. And today what I want to talk to you guys about is I want to ask the question, are these ecological effects evolution the results of direct interactions between community members, or is there a role for indirect effects of evolution? And this is an important question because whether or not there's a role for evolutionary indirect effects in changing ecology can alter the predictions that we may make as a function of that evolutionary change. The study system that I work in is with Trinidadian guppies, this little, wrong button there, small little fish right here. And Trinidadian guppies on the Caribbean island of Trinidad live in 2 communities. Below barrier waterfalls, they live with Hart's killifish, this guy right here, 2 species of fish predators, and then several other fish in the community as well. Above these barrier waterfalls, where these other fish are not, they live only with killifish. These differences in the community types where guppies live drives evolutionary changes in guppies between these 2 sites. And there's a very long history of studying genetic variation in guppy traits, including things like the life history, demography, behavior, morphology, and swimming performance. A few years ago, we published a paper on whether or not this local adaptation in guppies influences ecosystem dynamics, and this is just a small part of the results from that paper, showing that if you have guppies coming from high predation localities, these triangles in red, that mesocosms where we have them housed in tend to have fewer invertebrates, but then also more algae, as you see over here. So there's a very strong signal of changes in the phenotype having community effects. So what I want to talk to you guys about today in relation to guppies specifically is, are these observed effects that we see in these types of experiments driven by direct interactions, how the guppies are interacting directly with the ecosystem, or is there something going on through these indirect effects? And as Joe mentioned in his introduction, I'm going to talk to you guys about some experimental studies in mesocosms where we use electrified exclosures to separate direct versus total indirect effects. But then also I'll tell you a little bit about some mathematical modeling that we've been doing with a colleague, Régis Ferrier, on modeling the ecosystem fluxes within these communities to decompose that total indirect effect into multiple constituent parts. This is the setup of our mesocosms that we have in Trinidad. We have 16 mesocosms that are set along a natural stream, so the mesocosms get all of this sort of influx of invertebrates and whatnot that occur in the stream. We can set up replicated factorial designs. So in this case, for this experiment, we crossed the phenotype of the guppies with density, an traditionally important ecosystem effect. And then also we have some treatments where we don't have any guppies, which I'm not going to talk to you guys about today, but you can talk to me about it later. Because we can do these experiments in a replicated way, we are able to do this with multiple independent origins of the phenotype, so we can gain some generality about whether or not these truly are a function of evolution between the two stream types. Within each mesocosm, as I mentioned, we can set up electrified exclosures, wherein what we can do is we can keep guppies from having access to some parts of the mesocosms and allowing them full access to the others. And what that means is that in the areas where guppies don't have access, the only way that guppies can influence benthic algae is to influence them through their indirect effects. So we can measure, quantify indirect effects of the guppies on something like algae by measuring changes there. And then outside of the electrified exclosures, the mesocosms are influenced by both the direct and indirect effects, and so we can separate those statistically by having a design like this. So we can prepare the mesocosms, seed them with invertebrates, make them look pretty much like a natural stream. We can release fish into them, and then over a 28-day period, we can measure changes in the ecosystems and the communities that are occurring as a function of those different treatments. We can come back, we capture the fish, and then spend a lot of time and effort processing samples and analyzing data, of course. This is the model of the ecosystem dynamics that occur inside the mesocosms. And this is, although there's lots of equations here, it's not all that difficult to understand. This is your basic sort of food web flow diagram. And then you've got fish up here. This would be the guppies. These are our pool of invertebrates, periphyton, and then nutrients and detritus. And the system is described by these series of linked differential equations, which each one is a function of these different flux rates between these different compartments. And most of these fluxes we're able to measure. A lot of these fluxes we're able to measure during the course of the experiment. Some of them are parameterized based off of independent studies that we've done in Trinidad, and then some of them are taken from literature values. So on to some results. So what you're looking at here are this gray bar here, the one farthest on the left, is the total effect in terms of guppies on algae, which is the high predation minus the low predation. So it's the difference in how much algae is in the streams in high predation versus low predation. And so positive values here mean that the total change between the phenotypes is positive, which means there's more in the high predation channels. Okay, so, and then what we can do is you look here, these are the direct effects. So this tells us what component of this net effect is due to direct influences, which is basically for algae is guppies grazing on algae. The red bar, the farthest one to the right, tells us the total indirect effects. And so indirect effects can be things like guppies excreting nutrients and influencing producers through fertilization effects, or trophic cascade effects. And if you remember, I told you before that high predation guppies eat more invertebrates and they eat less algae. They also excrete nutrients at higher rates. And so any one of those different pathways should have led to there being a positive indirect effect in the context of this experiment. But you can see that the indirect effect not only is smaller than the direct effect, but it's also negative. And so this puzzled us for a little while. But as you remember, I told you we have this ecosystem model. And so with the ecosystem model, what we can do is instead of just looking at the total indirect effects through exclosure studies, we can break down this small negative indirect effect into its constituent parts. And when you do that, you can see that this bar here represents the indirect effect simply going through a trophic cascade. So there is actually a very large indirect effect due solely to the trophic cascade. If we toggle through here, you can see that this is what I'll call a nutrient cascade, which is the effect of high predation guppies excreting nutrients and then fertilizing, causing producers to grow more. And that effect is even much larger than their effect on the trophic cascade. But the really neat thing to show you here is that even though that's the case, if we look at higher order indirect effects, second order indirect effects, so guppies eating invertebrates, and then the loss of the nutrient recycling due to the invertebrates because they've been eaten, actually has a much larger effect and an effect that's opposite in sign to what we see with other indirect effects. So the sum of these indirect effects here, these ones, gives you this total direct effect, which is why it's negative. And it's because of this very large effect of lack of nutrient recycling in the invertebrates. Or at least that's what the model tells us.